tiene con nosotros desde el día de ayer con los chicos de la licenciatura. Stewart obtuvo su bachelor en biología en el College Swarthmore. Después hizo su doctorado en Stanford, trabajando ya desde ese entonces con genética y morfogénesis en Arabidopsis en embriones. Y después hizo un primer postdoctorado en el International Maize and Wheat Improvement Center, para después hacer un segundo postdoctorado en la Universidad de Pensilvania hasta el 2009, y en el 2010 se mueve a México a Langebio, al CINVESTAV en Irapuato, y desde entonces ha estado en México trabajando en el desarrollo de Arabidopsis y la parte genética. Lo que me falta decir es que es investigador nivel 3D, que me imagino que es de lo más alto en el CINVESTAV, y es miembro del Sistema Nacional de Investigadores también. Ha publicado alrededor de 40 artículos científicos en revistas de muy alto impacto, dirigido más de 20 alumnos de posgrado y 4 postdoctorados. Ha impartido múltiples conferencias alrededor del mundo en diferentes foros donde se discute el desarrollo de plantas. Y también comentaba, y bueno, me consta que es un excelente profesor de genética y biología molecular. Ayer en la clase aprendí bastante. Entonces le doy las gracias nuevamente a Stewart por haber aceptado la invitación, y bueno, hoy nos va a hablar de el genoma cigoto, su activación en el cigoto y el padre, las funciones en la embriogénesis de Arabidopsis. Gracias, Stewart.
Okay. Muchas gracias. Pues voy a impartir mi seminario en inglés, pero hablo bien español, entonces si tienen preguntas me pueden preguntar en español, y también me pueden interrumpir si quieren, si no entienden algo. So I was looking back at in my old folders of my old seminars, and I saw that it was 2012 the last time I gave a seminar here at the IBT, and I thought the auditorium looked different, and in fact it does. So I'm going to talk about a project that is the main project in my lab that I was just starting, and we had just the first exciting results back in 2012, and now we thankfully have a lot more results. So we work in my lab mostly on Arabidopsis and on Arabidopsis seeds, and mostly on embryos, because it's a very interesting developmental system. People have been fascinated with embryos and developmental biology for hundreds of years, because you start with a single cell and that makes a whole organism, and so it's really an amazing process. And in plants, most people that work on embryogenesis work on Arabidopsis, and because of the advantages of Arabidopsis, and also because it's so easy to see the embryos. So this obviously is a flower. Here's a silique, which is the seed pod. If you open this under a dissecting scope, you see the developing seeds. And because Arabidopsis only has a transient endosperm, it's very easy to clear the seeds and look at the embryo. You just clear with a protocol similar to how Drosophila embryos are cleared, and use Nomarsky optics. And you get these beautiful images. And seeds are also interesting because they have different genetic components. So the seed coat is maternal it's a maternal sporophytic tissue. It comes from the mother plant. The endosperm, which is the nutritive tissue, analogous to the placenta in mammals, is triploid. It has 2 maternal genomes and 1 paternal genome. The embryo is diploid with 1 maternal and 1 paternal genome. And a lot of the research in my lab is directed towards trying to understand the relative contributions of the maternal and paternal genomes in early seed development. And so we can clear them. We've done thousands or millions of these clearing preps, probably thousands, and it's a very simple protocol, and you can really see a lot. So that's why Arabidopsis is so good for studying embryogenesis. So I'm going to talk about the maternal to zygotic transition, in particular zygotic genome activation. So the maternal to zygotic transition has been studied in animals for quite a while. It's basically the transition from regulation of embryogenesis from products from the egg cell, so proteins and mRNAs inherited from the egg cell and the zygote, transitioning to zygotic regulation of embryogenesis. And in animals, people have studied this for a long time. The timing is a little bit different, but basically you have this beginning period where there's these maternal transcription proteins regulating development, and then the zygotic genome activates, you start zygotic transcription. And there's various hypotheses about why there would be maternal control of early development. It depends a little bit on the species. One is that if, for example, in Drosophila, the nuclei are dividing so quickly that you can't transcribe anything but very short genes because nuclear division is so fast in Drosophila. There's also the idea that gives you some time to reconcile epigenetic marks from the gametes. There's also the idea that it might avoid too much paternal control, the parental genome conflict theory, where, for example, especially in outcrossing species, the mother in plants, it would be the mother plant, all the seed are genetically related to the mother, whereas if it's an outcrosser, the pollen might be from different plants, and there's this idea that the paternal alleles might try and promote resource allocation to their seed and be in competition. And there's other hypotheses you can come up with. So when I started this work more than 10 years ago, so here's the general idea in animals: early embryogenesis, maternal control, then you transition to zygotic. There were kind of two competing camps for how this worked in plants. One is that it was similar to animals, where there was initially maternal control of embryogenesis. And there was another school that thought, no, it must be zygotic control almost from the very beginning, that there's very little maternal regulation of embryogenesis. And so that my talk is going to focus on experiments we've done to try and understand what's going on here. And there's 4 parts of my talk. I'm first going to talk about zygotic genome activation in isogenic embryos. Okay, and so one of the points of my talk is that a lot of the confusion is because when you use hybrids to study parent of origin transcriptomes, different hybrids give different results. And so we think that's a lot of the reason why there was controversy in this field. But first I'm going to talk about studies in isogenic embryos, then zygotic genome activation in hybrid embryos, then some meta-analysis we've done of transcriptomes and epigenome data, and then finally a little bit of very new results on the function of paternally biased genes in early embryos. So these are transcriptome studies in maize, rice, and Arabidopsis, comparing transcriptomes from the egg cell and a very early zygote. And there are serious transcriptome differences as little as a few hours after fertilization. So I think pretty much the whole field now agrees that yes, transcription in the zygote of at least a portion of the genome starts quite early. There's no delay of a couple days. In each of these cases, it's about 10% of the genome transcripts increase in the zygote compared to the egg cell. But there were results starting from 2000 where using enhancer traps, so these are markers for a transgenic marker for different alleles, using crosses from the father, the transgene, in this case Gus, would not be expressed for the first couple days after fertilization, but if you pass the transgene from the mother, it was expressed immediately. So it looked like there was some kind of paternal silencing. This is not inconsistent with this result where there's early transcription, but it suggests that maybe this transcription might have a maternal bias initially. Okay, so this is in 2000. Here's a later study. It was more carefully done with much more counts. And it's not that these are different markers. It varies between genes. Some have almost no paternal signal, and others just have less paternal signal than maternal signal. So some kind of paternal silencing, but it is not complete or less paternal activation. This idea that there might be a paternal transcriptome, maternal transcription bias was very unpopular. And I always struggle to understand why. So here's an article just showing a marker line which was supposedly equally expressed, not quite true, but more or less equally expressed from both alleles. But I think a lot of the resistance was from genetic data that if you look at embryo defective mutants, so there's about at least 500 different genes in Arabidopsis that are required for embryogenesis, and so mutants have these phenotypes very early on. You're going to see a lot of them. And if you look at the if you take a plant that's heterozygous for an embryo defective mutant, okay, and you look at the progeny, so if it's, you'd expect that 25% of the seed would be mutant if it's a zygotic effect. And that is what you see for almost all embryo defective mutants. This is a silique that I opened. Here are green normal seeds. Here are white seeds, and they're white because the embryo is tiny and doesn't have chlorophyll, or it's just dead and tiny. So if there were a strong maternal effect, as suggested by these Gus staining studies, you would expect that you would see more like 50% mutant embryos, because if the maternal allele is more important. But that's not what you see. So there was quite a bit of resistance to this idea of a maternal bias in early embryogenesis. So when I was starting my lab, we wanted to work on this subject, and one of the first things we did was order probably about 100 embryo defective lines from the stock center. And my master's student at the time, Gerardo del Toro, did a very careful study looking at the segregation ratios of phenotypes starting very early, so starting about 2 days after pollination. And it turns out, if you look in late siliques, like this, yes, they segregate about, this is a heat map where more purple is higher percent mutants. If you look in late siliques, yes, they segregate about 25% mutants. But if you look very early, you actually see, in some cases, closer to 50%. So there is a maternal effect for some of these, but it's transient. So if you don't look early, you won't see it. Okay? But there's another kind of experiment you can do, genetic experiment that's a little bit easier to interpret, and that's just if you use a heterozygous mutant as a mother or a father and cross it with wild type as a mother or a father. And that way you can check for maternal effects or paternal effects. And a maternal effect is when the phenotype of the embryo depends on the genotype of the mother, whereas a paternal effect is when the phenotype of the embryo depends on the genotype of the father. And so we did this kind of assay. Initially, Gerardo del Toro did this for about 50 different mutants. And here it is, the two different scenarios looking for maternal effects. If you cross a heterozygous mutant as a mother with wild type as a father, if there is no maternal effect, all the embryos are going to look normal. But if you have the kind of transient maternal effect that we saw here in self plants, what you'll see is initially you'll see mutant phenotypes like here, but their percentage of mutant phenotypes will gradually decrease until there are almost none, because plants are amazingly plastic and can recover from embryo defects. And so Gerardo did this for 49 different embryo defective genes, and we found basically 2 different classes which are represented in this heat map. There were 9 genes for which he really didn't see any maternal effects. So the conclusion here is the paternal allele is transcriptionally active from the very beginning, and the paternal allele can complement the lack of a functional maternal allele. So there's 9 here. But the majority of the mutants showed transient maternal effects with different penetrance and different expressivity, from ones that showed close to 50% to ones that had maybe 10%. And then as you go, this is 2 days, 3 days, 5 days, 14 days after pollination. In all these cases, the embryos eventually recover and you have pretty much completely normal seeds, even though the mother is mutant. And here's just some examples. So these are clearing preps. Here's wild type. There's a 2-cell embryo, a globular embryo, and a heart stage. Here's an example of where the paternal allele is immediately functional. There are no maternal effects. These mutants are actually heterozygous for ATSY3A, but you don't see anything. Here's a control of a self plant. And here's another, the more common case, when the mother had a mutant allele, the father is wild type. You see mutant phenotypes early on, but then you see fewer and fewer as development goes on. And more recently, my master's student Axel Orozco tested even more mutants, and he tested maternal and paternal effects specifically. And again, purple is when you have a maternal effect or a paternal effect. And he found that of these 24 mutants, they all showed some kind of maternal effect, but only a few had a paternal effect, and the paternal effects were less than the maternal effect. So from this first part of my talk, the conclusion is that yes, transcription of the genome does start soon after fertilization, but it's only part of the genome. So there is early, but not complete, early zygotic genome activation. And maternal alleles of embryo defective genes are definitely, in general, more functionally important than paternal alleles. But if you want to look at transcription, you need to do crosses between different varieties of Arabidopsis so that you can use the SNPs to call the reads as coming either from the maternal or the paternal genome. So here's an example of Landsberg and Columbia. They have a polymorphism at this base pair here. You cross it in the zygote, it's technically difficult, but you can sequence the transcriptome of zygotes and early embryos, and you just assign the reads back to the maternal or the paternal genome and look to see if you have more maternal or paternal transcripts. And so there were a few studies that were done. One in 2012 using the Columbia-CVI combination, crosses in both directions. At the earliest stage was the 1 to 2 cell, and then the globular stage. This study found equal maternal and paternal transcripts at the genome level. Then there was a later study, also in Arabidopsis, using the Columbia-Landsberg hybrid, where they found a very transient maternal bias. So at 14-hour after pollination zygotes, they found an excess of maternal transcripts, but in the 24-hour after pollination zygote, there were equal maternal and paternal transcripts. Meanwhile, studies in rice found something different. They found that there was a massive maternal transcript bias. And in rice, they can dissect, zygotes are very easy to dissect, or easier to dissect and to time exactly. And so they found that yes, at 2.5 hours after pollination and 9 hours, there's a massive maternal transcript bias, like 98 or 99% maternal transcripts. So these different results, different species, different hybrids in the case of Arabidopsis. But in Arabidopsis specifically, this equal maternal-paternal transcript contribution really doesn't make any sense with our functional results in isogenic Columbia, because if all genes had equal maternal and paternal transcripts, you wouldn't expect to see any maternal effects except in the case of haploinsufficiency, which we saw a little bit, but not very much. So this didn't really make sense. We were scratching our heads about this for quite a while. There's a lot of controversy about this. So we continued doing our genetic experiments, and we did them with hybrids. Okay, so now we used our embryo defective mutants in Columbia and crossed those Columbia mothers with plants of these different varieties, the ones that they used for these transcriptomes, right? And we got a very nice result, which is consistent with the transcriptome studies. This is maybe a little bit, so this is our functional data. These at 2, 3, and 5 days after pollination. Again, the number of mutant phenotypes you observe when you cross an embryo defective plant heterozygous as a mother with different ecotypes as a father. They've been normalized to the number of mutant phenotypes you see when you do isogenic crosses, okay? So in using CVI and Landsberg as the father, you see less maternal effects, which is totally consistent with these hybrids showing earlier paternal transcripts. So that was a nice result. And we've had these other two hybrid combinations using Tsu as a father or Burr as a father, where statistically there wasn't any difference in the maternal effects. And so these hybrids looked like more interesting hybrids to use if you want to understand parent of origin behavior in isogenic Columbia, because they functionally are the same as isogenic Columbia. And during the pandemic, my former PhD student, Jaime Alanis Fabian, we were also wondering, well, how he started digging into these transcriptomes more. He went and downloaded the raw data and reanalyzed it himself. And he found some interesting things. So here he's representing the data as the original authors represented it, which is a log2 fold chains ratio of CVI reads over Columbia reads. So in this case, when the log2 fold change is 0, it means there's equal maternal and paternal reads. And so you get this nice curve here with a spike in the middle. So that was, for Columbia CVI, almost all reads are equally distributed between maternal and paternal alleles. In Columbia and Landsberg, at 14 hours after pollination, you get a slight shift to being maternal, but then in 24-hour after pollination zygotes, equal maternal, paternal, also in the apical cell and the basal cell, and also in the globular embryo and suspensor. So Jaime remapped all these reads, and he represented them not as log2 fold change, but as something much simpler, just as maternal fraction. So a maternal fraction of 1 means that all of the reads were from the maternal allele, and a maternal fraction of 0 would mean all the reads are from the paternal allele. So for the CVI Columbia hybrid, he got essentially this same result as the original authors, which is that they're all clustered around equal maternal and paternal reads with very small spikes here. But he got a really different result with the Columbia-Landsberg transcriptome. You got these really strong peaks here at completely maternal and completely paternal in zygotes at 14 and 24 hours after pollination. Very interestingly, in this combination, for the first division of the zygote, the apical cell starts to show reads that are equal maternal and paternal, but you still see this parent of origin behavior persisting in the lower cell, in the suspensor cell, the basal cell. And you still see the behavior a little bit still in the globular embryo. So this explains, so this is one of our very important result, that actually this study, which claimed that there were equal maternal and paternal reads, the data that didn't agree with our hypothesis just were not present in the analysis. And so to dig in a little bit more, this is just looking at Columbia-Landsberg zygotes at 14 and 24 hours after pollination. It turns out there are quite a few that are biallelic, but you also have many that are unidirectional. So that's where they're highly biased in one direction of the cross, but not biased in the other direction of the cross. So these early Columbia-Landsberg transcriptomes are much more complicated than initially proposed. And so why were our results so different than the original author's results? There are two reasons. One, so Jaime added pseudocounts in the case where you have a gene where the reads are either all maternal or all paternal. He added a few counts in the other direction so that, and it's common practice apparently in bioinformatics, so that you don't get these log ratios that just go to infinity. And when you just look at the data visually, it just, when they're represented as log 2 ratios of one ecotype over the other, it gives you something like this. But if you show the same data as maternal fraction, exact same data, you can actually see these strong paternal and maternal peaks. So just representing data as log 2 fold change tends to emphasize visually genes which are in the middle and de-emphasize ones which have extreme parent of origin behaviors. And so we, since quite a while, had a collaboration with Raju Datla's lab in Canada, and they had actually done a transcriptome with Columbia 2 hybrids, the hybrid that we think is more representative of isogenic Columbia. And they shared their data with us, and we analyzed it. And as expected from our functional data, yes, there is a very strong maternal transcript bias in zygotes and at the octant stage, which moves to being biallelic at the globular stage, which is exactly what we would expect from our functional data, where there's strong maternal effects early on, but then by the globular stage at 3 days after pollination, they're almost gone. So this is a really nice result. So one initial problem in this field is that if you have contamination, it's very hard to dissect zygotes and early embryos from the seed coat, which is completely maternal. So if you have contamination of a little bit of either maternal tissue or maternal mRNA, it can skew your results. And so we were concerned about this. So to see if this were affecting our results, Jaime did this analysis where he compared the maternal fraction of genes on a gene-by-gene basis and contrasted it with the zygote to ovule ratio of expression of the gene. So here are genes that are expressed much more highly in the ovule compared to the zygote, and here are genes that are expressed much more highly in the zygote compared to the ovule. And if you had significant contamination of maternal tissues in this transcriptome, genes which were more highly expressed in maternal tissues, you would tend to be more maternal, but there's no correlation. So we really don't think that these results, we really believe these results. We don't think they're due to maternal contamination. He saw the same thing when he looked at the octant embryo data and compared it with the ovule. So, and here's another way of visualizing these results as dot plots. So the position of the dot shows the maternal fraction in both directions of the cross, when Columbia is the mother and Sue is the father, or when Sue is the mother or Columbia is the father. So at the zygote to one cell stage, There are thousands of genes with a very strong maternal bias, statistically significant. At the octant, so this is about 24 to 30 hours after pollination. At the octant stage, which is about a little bit more than 2 days after pollination, you still have a strong maternal bias, but it's less. So gray is no bias. In purple are the paternally biased genes, and yellow and green are genes that show allele dominant behavior. At the globular stage, this maternal bias is almost gone. Okay, so it's a transient maternal transcriptome bias. And another very interesting experiment Jaime did was to look at the maternal fraction of the gene in the zygote or embryo and compare it to the level of expression of the gene in the embryo versus the egg. And again, so these are the embryo to egg ratios binned, I think in 20 different bins. And there was no correlation with the maternal fraction of a gene and its expression in the egg cell versus the zygote. And so if we think that these maternal biases we see here are more likely to be due to maternally biased zygotic transcription and not actually inheritance of maternal transcripts. Because there's no if they were due to inheritance of maternal transcripts, genes which are highly expressed in the egg cell would tend to be more maternal than genes which are highly expressed in the embryo compared to the egg cell, but we see no, there's no correlation there. And then he did another really interesting experiment that we don't have data for that many genes because he looked at intron reads. So these are reads from transcripts just after they've been transcribed when they're still in the nucleus, okay? So they should represent active transcription and not transcripts that have been inherited from the egg cell. But since most transcripts are processed very quickly, you don't get intron reads for that many different genes, but he has them for a few hundred. And it turns out that at the zygote and octant stages, the intron reads here are in blue, they're highly, highly maternal, but by the globular stage, they're not. So this is pretty good evidence that we think that it's actually maternal zygotic transcription and not inheritance that's giving this maternal bias. So conclusions from this part, Col2 and Col-Burr hybrids functionally are indistinguishable from isogenic Columbia. Col2 hybrids show a maternal transcript bias in early embryos. We have some evidence that this maternal transcript bias is really due to maternal zygotic transcription and not inheritance of transcripts from the egg cell. Columbia-CV hybrids show essentially no parent of origin behavior as was originally found, but Columbia-Landsberg hybrids show extensive monoallelic expression in the zygote, which after the first division transitions to being biallelic. So these hybrids are much more complicated than originally proposed. Now I'm going to get into some meta-analysis of transcriptome and epigenome data where we're trying to contrast these different hybrids and try and understand a little bit mechanistically what might be going on. So here's the same data that I've been showing you, but analyzed all the same way. These number of genes here, Columbia 2 and 2 times Columbia data, they have this strong maternal bias at the zygote, one cell, and octant stage, and later they're equally distributed. Columbia CVI, so it's essentially no bias. Columbia Landsberg initially has these strong maternal and paternal peaks, but after the first division of the zygote, the apical cell is biallelic, okay? So why would this be? Well, you think it probably has something to do with epigenetic marks, either in the gametes or that are then remodeled in the zygote, maybe remodeled differently. There's some evidence about epigenetic regulation of paternal gene expression. This is experiments with Gus markers from the Auditorio et al. Paper, where they found that the mutants in the maternal RDDM pathway led to earlier or higher paternal expression. So some evidence for transcriptional silencing of paternal alleles, maybe in the embryo. Other interesting evidence, CVI turns out to be the outgroup in this study of more than 100 different ecotypes. CVI has almost no CG methylation. And so CG methylation, of course, has a lot to do with transcriptional genome silencing. And so Jaime did this analysis here where this is comparing our functional data for these embryo defective mutants crossed with different hybrids with differences in CG methylation between Columbia and these different ecotypes. And there's a statistically very significant, but not super strong correlation between genes which have less CG methylation, showing smaller maternal effects, and genes that have more CG methylation tending to show less maternal effect. Okay, so that's generally consistent with CG methylation having something to do with perhaps silencing the paternal genome. And then these are more recent results where here he's graphing the maternal fraction of different crosses. Okay, so this is in zygote 1 cell, the Columbia 2 cross, and this is the maternal fraction, and these are the number of genes. And so here, this blue line, these are genes which are almost entirely maternal. Here are genes which are very, very highly paternally transcribed, and here are ones which are kind of in the middle. And these two peaks, the highly maternal and the highly paternal, are correlated significantly as having less CG methylation. This is methylation in leaves, than these genes here in the middle. So, and the same in the other direction of the cross. So there's an interesting correlation that less CG methylation leads to stronger parent of origin transcription. When we looked at the Columbia-Landsberg hybrid, at 14 hours after pollination, when there are these very strong maternal and paternal peaks, we saw a similar thing. These highly maternal genes and highly paternal genes tend to have significantly less CG methylation than genes that are in the middle in both directions of the cross. And also we saw the same thing when Columbia is the mother in a Columbia-CVI cross. This is a little bit more tricky because CVI has very little CG methylation, but we also saw a good correlation between genes that are highly maternally transcribed. They tended to have less CG methylation than the rest of the genes. So preliminary conclusions from this: lower CG methylation is associated with genes that show extreme monoallelic behavior. It's not associated with these genes that were like in the middle. These are maternal biased, but not so maternally biased. So suggests there's probably different epigenetic mechanisms going on here to regulate parent of origin gene expression. Maybe the RDN pathway, maybe polycomb. We're looking at that now. So this is a model we proposed years ago in a review, which we think is more or less correct. So here, in contrasting what goes on in isogenic embryos versus hybrid embryos. So isogenic embryos, we think in green is the maternal genome, purple is the paternal genome. For most but not all genes, there is paternal silencing in early embryogenesis, but some genes do not show this paternal silencing. And when you look at hybrids, they behave differently. There are some hybrids like the Columbia 2 hybrid, which seems to be similar to isogenic Columbia. There's other hybrids, so that's in orange here. There's other hybrids like the Columbia-Landsberg hybrid, where the genome activation varies a lot between genes. There's others where there's not much difference in activation of the maternal and paternal genome, where you have very similar maternal and paternal allele reads. And again, this is just going back to the data. CVI doesn't really show any parent of origin behavior. But here, looking at Columbia-Landsberg, you do see this maternal peak here. This is in 14-hour zygotes, which goes away by 24-hour after pollination zygotes. And just looking at it, it looks similar to what we see in Columbia 2, but obviously the timing is very different. Here in this Columbia-Landsberg hybrid, this maternally biased transcription in the zygote is very transient. And here in this other hybrid, it lasts a couple of days instead of just a few hours. So that's our working hypothesis, that there's actually, it's kind of a combination between these two original models. There is a maternal bias in early embryogenesis, but it's actually zygotic transcription bias, not due to inheritance of maternal transcripts, and it varies in different hybrids. And just the last part of my talk, I wanted to mention some functional studies we're doing now to try and see if these biased genes have any functional relevance. So we're looking at doing a couple different things. So here, going back to the Columbia 2 transcriptomes, there's a small number of genes that show a statistically significant paternal bias, right? So they're paternally biased regardless of the direction of the cross. And we thought, okay, well, there's not very many of them. We wonder if these paternally biased genes are paternally biased because they're paternal transcription is actually functionally important, and a way you can do that is just looking at mutants in these genes. So we've looked at, so far, mutants in 9 different of these paternally biased genes. Okay, so again, these genes here. And for 5 of them, we found mutant phenotypes, interestingly, primarily affecting the suspensor. Okay, so this is, and most of these genes have not been characterized. This gene and this gene haven't been characterized at all. This is a transcription factor, which has been characterized a little bit. This is a polycomb motif, no information here. And this is either a microRNA or another transcription factor, which I'll get to in a minute. The penetrance of these phenotypes is not super high, but it's not super low. But they all affect the suspensor, which is really quite interesting because, again, these are paternal effects. So these are genes which normally are promoting growth of the suspensor. And the suspensor, one thing it does is to push the embryo into the endosperm tissue where it can get more nutrients. So this is an example of maybe paternal alleles trying to promote nutrition of the embryo. So this mutant in this FBOX protein has altered divisions in the hypophysis, short suspensor, and they're slowed down, short suspensor and fewer divisions. And this microRNA or transcription factor, which I'll get to in a minute, it has short suspensor and short cells. Besides that, it looks fairly normal. We're quite excited about this one, but it's a little bit tricky, and we're trying to figure this out now. So this microRNA, MIR5015, just upstream, probably in its regulatory region, it has this transcription factor, and we've looked at tDNA insertions in the microRNA transcript region and in the transcript region of the transcription factor, and they all give the same kind of phenotype. So we're trying to figure out if the phenotypes are due to the microRNA or the transcription factor. That's work in progress. I'm hoping that it's the microRNA, but it could be either, and anything is fine. We're particularly excited about the microRNA because, and here's just some statistics showing that the suspensor of the mutant is really about half as long. This is the mutant here in gray, wild type in purple. The suspensor really is about half as high in the mutant compared to the wild type. So it's really quite a clear difference. So our hypothesis, which is a really hypothetical hypothesis at the moment, it's known that MIR5015 is very highly transcribed in sperm. There's a study on that. We think that it's probably transcribed in the sperm and not in the zygote, so it actually shows parental transcript bias because it's inherited from the sperm cell. One of its targets is IN3, which is a ethylene responsive transcription factor, which tends to repress cell elongation. So one hypothesis that we want to test is that actually what's going on here is that this transcript is inherited from the sperm cell in the zygote, where one of the things that it does is to repress this transcription factor, which represses cell elongation, so the suspensor elongates normally. So we're following that up. It's a tantalizing result. And that's work from a master's student of mine, Paco Aguirre. And then my PhD student, Karina Orozco, has been working now for several years in her master's and now in her PhD on a different question, on a couple things actually. So we're wondering, we have good evidence that in the embryo, there's silencing of the paternal genome for a few days after fertilization, and we're wondering if this occurs in the endosperm as well. So as a kind of a preliminary experiment, Karina went and reanalyzed endosperm transcriptome data. It's a little bit too late probably to observe this paternal genome silencing because at least in embryos at 4 days, it's mostly over. So we're working on more experiments to generate our own data. But she did find that there were quite a few paternally biased genes in the endosperm at these 2 time points. And in her PhD, she's been following up on some of these genes. And one class of paternally biased genes that really caught our eye were the VIM genes. These are epigenetic regulators that link CG methylation, DNA methylation with histone methylation. Okay, and there's a very strong correlation between CG methylation and different kinds of histone methylation in these mutants. So this is in the endosperm. So if there were equal maternal and paternal allele transcription, you'd actually see 66% maternal because there's 2 paternal genomes and 33% paternal. And all these 4 VIM genes show a lot more paternal transcripts than you would expect. So there's some kind of imprinting or there's there's a bias towards paternal transcription. This is just a little diagram with the Vim genes. They basically link MET1 CG methylation with different kinds of histone methylation to maintain gene silencing. There's several hundred genes that change in these mutants in vegetative tissues compared to wild type. And so looking at their transcript levels from different published transcriptomes, they're all transcribed in the embryo and the endosperm except for Vim5, which is almost not present in the embryo at all, but is quite high in the endosperm. And so perhaps consistent with this, in Vim5 single mutants, we see this very interesting phenotype where the chalazial endosperm is increased compared to wild type. Okay, and so many of the nutrients that enter the seed actually pass, so that the vascular continuity with the mother plant is here. And so the nutrients that come from the mother plant into the endosperm have to pass through this chalazial endosperm. This is quite interesting that a paternally biased gene is regulating the size of the chalazial endosperm. It's the penetrance of this mutation is really quite high. So we think we're really excited about this result. And as far as the embryo, she saw at lower but still significant frequency in all these single Vim mutants, Vim 1, 2, 3, 4, 5, and 6, phenotypes where in general the suspensor was shorter so this, here's wild type, here's a mutant. So here's the wild type suspensor, mutant suspensor. In general, the suspensor in these mutants is much shorter than in wild type. And when Karina made a Vim 1, 2, 3 triple mutant, which took a little bit of work because Vim 1 and 2 were very closely linked, but she made this triple mutant and she saw much stronger defects in the embryo, abnormal divisions in the embryo proper, in the suspensor, shorter suspensor, this kind of club phenotype, reminiscent of an auxin phenotype. So we're looking into that. Also defects in the endosperm where some of the endosperm nucleoli are very big, others are very small compared to wild type, okay? And she did parent of origin tests to see if this was a maternal or paternal effect, and she found maternal and paternal effects of equal maternal and paternal effects, suggesting that it's actually haploinsufficient, that you need both copies of these genes to fulfill their function. So that's the end of my talk. Here are the people that did the work. Gerardo del Toro started this project. Axel continued with the functional analysis and with embryo defective genes. Jaime did all the computational biology analysis that I showed you of all the different transcriptomes and the epigenomes. This is Karina, who's working on the Vim genes and parent of origin regulation in the endosperm. Paco is working on the paternally biased genes in the endosperm, and I'll be happy to take any questions.
¿Preguntas?
En inglés o español.
Hola, perdón por mi voz. En español, porque dijiste. Tengo dos, no sé si lo mencionaste, pero de los 2000 genes más o menos que ves biased de materna, o sea, maternos, ¿hay algún grupo o vía particularmente enriquecida en estos genes?
Sí, no hemos visto eso. Checamos un poquito y no hemos visto alguna correlación biológica. Los genes que sí tienen sesgo materno están en todo el genoma. Eso no es el asunto. Una hipótesis podría ser que están más cerca de transposones, que hay silenciamiento ahí, pero no hemos visto eso tampoco. Es muy buena pregunta y no tenemos evidencia que hay algún go-term que está silenciado más que otro.
Y la otra pregunta es si tienes alguna idea o sugerencia de qué mecanismo puede estar usando la maquinaria basal para distinguir el alelo paterno del materno y agarrar uno.
Sí, pues probablemente es algo epigenético o metilación o del ADN o de las histonas o histones diferentes, que hay diferencias en los gametos, en la célula huevo, en el esperma, que tienen marcas diferentes, y en el cigoto estas marcas diferentes dan diferentes respuestas. Sí, pero no inicialmente. Y también otra cosa es que ¿por qué es diferente en diferentes híbridos? Podría ser que los epigenomas son muy diferentes. Pues ya son muy diferentes. Por ejemplo, CVI casi no tiene metilación CG. Otra podría ver con el intercambio de histonas que, por ejemplo, en el caso de Columbia-Landsberg, después de la primera división del cigoto, la célula apical, ya la transcripción es aparentemente igual entre los dos alelos que no era antes. Entonces quizás hay una histona que viene del esperma que impide la transcripción, pero hay reemplazamiento de las histonas durante la primera división y por eso cambia el patrón mucho. Si no sé, probablemente hay varios mecanismos y probablemente es diferente entre diferentes ecotipos y nos interesa mucho entender eso.
Les recuerdo a las personas que están en Zoom y YouTube que pueden escribir sus preguntas y aquí las leemos.
Gracias, Stuart. Muy interesante plática, como siempre. Pareciera ser entonces que los efectos paterno-materno, no hay una tendencia de uno o en otro en realidad, ¿no? Que depende mucho de qué genomas usas en una cruza. Y sobre todo cuando dices que el mecanismo, o cuando los datos sugieren, pues, que el mecanismo es epigenético. Entonces me llama la atención, por ejemplo, en el caso de la CBI, con la cruza con la CBI, que es muy parecida a la isogénica. La CBI no tiene metilación CG.
No, CBI es muy no parecido al isogénico.
Es la que no es
la
parecida.
Más diferente, sí.
Entonces sí checan
sí.
el que no tenga metilación CG.
Sí.
Entonces digamos que podríamos decir que es clarísimo que, pues lo más claro, que sí está, que al menos en esas cruzas, en las isogénicas, sí está mediado por metilación tipo RDM.
Metilación CG.
CG.
Y también vimos cierta correlación entre el nivel de metilación CG en los genes en sí, entre los diferentes ecotipos y lo que vimos con las cruzas, con los ecotipos. Sí, metilación CG yo creo que sí tiene algo que ver, pero no entendemos exactamente cómo todavía.
Ya, y entonces, nada más viendo, a lo mejor en el modelito que tú pones en realidad es un gradiente más que, ¿no? Un gradiente muy sutil donde va influenciando más el genoma paterno en unos casos y en otros el genoma materno, ¿cierto?
Puede ser, sí.
Y esto lo han visto en algunas otras especies que no sean Arabidopsis.
Estamos ahora checando los datos de arroz, en donde hay muy buenas transcriptomas y también datos epigenómicos. En Arabidopsis usamos los datos de hojas, que creemos que está bien porque realmente la metilación no cambia tanto durante el desarrollo. Entonces creemos que las hojas sí probablemente dan resultados que aplicables al embrión, pero en arroz tienen epigenomas de células huevo, de espermas, de cigotos, y estamos checando, Jaime está checando eso ahora, y profesionalmente sí hemos visto, al menos con CG, también los genes que muestran o supermaterno o superpaterno tienden a tener menos CG en los gametos. Pero estamos, ojalá, pues sí vamos a publicar eso pronto, pero Jaime está analizando eso literalmente ahora.
Muy bien, muchas gracias.
Voy a leer una pregunta de YouTube. Víctor, alumno de la clase que estuvo por Zoom ayer, nos pregunta si es posible que el bias transcriptómica materno se haya originado como un mecanismo de defensa en los casos de las cruzas híbridas.
Sí, pues una hipótesis sería que yo quería probar desde mucho, pero no es tan fácil. Si la hipótesis de la guerra entre los genomas, por ejemplo, sobre todo en plantas que son outcrossers, ¿no? Entonces la idea del conflicto de los genomas de los padres es que si tienes una planta que es outcross, no sé cómo se dice en español, que es fecundado por muchas plantas diferentes, las semillas en estas plantas, todos tienen su genoma de la madre, entonces la madre está relacionada con todas esas semillas, pero tienen diferentes padres. Entonces, la hipótesis es que los alelos de los padres van a tener selección para tratar de tener más recursos de la madre, porque los alelos paternos que sí promueven más recursos de la madre van a hacer semillas más grandes, que van a crecer mejor, etc. Entonces, pero la madre, como tiene la contribución igual a todas las semillas, quiere impedir eso, quiere dar recursos iguales a todas las semillas, y por eso quizás hay silenciamiento del genoma paterno en embriogénesis temprana. Otra hipótesis de Uli-Gross-Niklaus es que este silenciamiento paterno es efectivamente una alargación del gamete ficto para dar más tiempo para seleccionar en contra de alelos detrimentales. Es otra hipótesis. Y si fuera cierto que funcionalmente si el genoma de la madre está silenciando el padre en Arabidopsis, que casi siempre hay autofecundación, hay muy pocas cruzas con otras plantas. Si checaríamos en una especie que es obligate outcrosser, uno pensaría que el fenómeno sería aún más exagerado, pero no es tan, sí es un proyecto muy grande que me gustaría hacer un día.
In English.
Okay.
Thank you very much. That's really interesting. I just want to understand better. Maybe I didn't understand. So your data on transcriptomic data in the second part, you showed that maternal bias transcriptome really becomes lower after third division, let's say in octant stage.
It decreases at the octant stage. It's less, but it's really over in the globular stage 3 days after pollination.
Okay, so this is my question is really, does it depend on genotypes, or it is a general effect that it goes just down as a result of cell division, or just normal development?
Well, I mean, so it's definitely different in different. So you need to use a hybrid to study this,
Okay.
unless you do it functionally with our embryo defective mutants in Colombia.
Okay.
Those functional results say that, yeah, there's the maternal effects, the paternal silencing lasts for about 3 days, and then it's pretty much over. It would be interesting to look at the embryo defective mutants in other ecotypes, which we did a little bit, and we saw similar things. But it's probably different in different ecotypes if you could test it, but you can only test it by doing these crosses. In the hybrids, yes, it really changes a lot. And again, we think that's probably due to epigenetics or other aspects of hybridization.
Okay. And another question about this WIM triple mutants. So these defects in suspensor, are they reproduced, let's say, in normal and in radical, let's say, in the quiescent center formation?
We haven't. So the VIM mutants, the more you self them, the sicker they get. I think I actually have no, I don't. Okay. I had a slide, but I got rid of it. So if Karina propagates them, they're segregating VIM3. If she propagated them as triple homozygotes, they get sadder and sadder every generation because, of course, you're getting less and less methylation or more and more altered epigenetic state with every generation. We haven't looked at roots. If you would like to look, I would be happy to give you some seed. The phenotypes that she sees in the embryo are consistent with auxin phenotypes. Well, auxin does everything super important, but you see that kind of phenotype in auxin mutants. We would like to learn more. We're going to do some crosses with some marker lines to see what it is.
Okay.
But yes, it probably has some effect on auxin at some point.
Yeah. Very interesting data. Thank you.
La siguiente pregunta de YouTube, que es de Daniela Delgado, es relacionada con la última, y pregunta que qué tanto es la marca sobre el DNA, las marcas de la metilación, o sobre la topología de los cromosomas.
Pues solo sabemos las marcas en el ADN o en arroz vamos a poder ver marcas en histonas. Pues obviamente las marcas en el ADN y histonas tienen mucho que ver con la conformación del cromosoma o cerrando o abriendo la cromatina. Entonces, pues no puedo decir más que eso, que sí, seguro que está también relacionado con la conformación de la cromatina.
No sé si han pensado o probado si mutantes en metilación o policombo o algo así pierden el bias.
Sí, hicimos un intento, creo que vamos a hacer otro. Uno pensaría que, por ejemplo, es muy buena idea, usando, si por ejemplo, si el problema con CVI es que, o la diferencia con CVI es que casi no hay metilación CG, ¿por qué no usamos la mutante MET1 en cruces con? Sí, como intentamos eso, pero vamos a intentar otra vez. Es muy buena idea y uno pensaría que sí, que tendremos los mismos resultados, sí.
¿Alguna otra pregunta? Bueno, yo sí quiero preguntar. Adelante.
At the level of transcription itself, do you think that you would have the machinery of the paternal genome that is working on the paternal genome and the maternal genome is working, and little by little you have domains that mix together? Or do you think that it's a question of conformation of the paternal versus maternal genome domains after fertilization?
I think it's more a question of cis of the maternal and paternal chromosomes and not the machinery, because they're there in the same nucleus, and we see this effect after multiple cell divisions. In Columbia 2, you see it when the embryo has like 16 or 20 or cells. So it's definitely not something that changes immediately, except in Columbia-Landsberg hybrid, it does, right? So after the first division, the apical cell is now transcribing pretty much equally, and the basal cell not. But the apical cell is super different in the Columbia-Landsberg hybrid compared to the zygote. But I would imagine it's more a cis thing to do with the conformation of the chromosome somehow, and not the machinery, because, well, although it could be, but I mean, they're there in the same nucleus, right?
I'm just an idea and just general question. If you know something about a recombination of maternal and paternal chromosomes, it's known that in somatic cells recombination do occur with very low level. I'm just curious if there are some studies about this on this paternal view, parental
Not
view.
Not that I know of.
Okay.
I don't have an answer to your question. Sorry.
Just curious.
No idea.
Okay.
Thanks.
¿Alguien más? Bueno.
Stewart, los análisis de la expresión de los transcritos que sean maternos o paternos y de lo que acabamos de discutir, implica que es a nivel de transcripción.
Sugiere, sí.
Bueno, sugiere que es a nivel de transcripción, pero ¿debería ser a nivel de transcripción necesariamente? ¿No podría tener un componente post-transcripcional en el cual
por supuesto, sobre todo en el cigoto, claro, sí, podría
los
ser.
transcritos paternos están marcados para
sí es posible. Eso podría explicar algunas diferencias entre los diferentes híbridos. ¿Es posible? Sí. Sí. Pues tenemos un proyecto en su infancia de ver la destrucción o la regulación de los transcritos de la célula huevo y cómo es que bajan rápidamente, etc. Pero sí, claro, podría ser alguna regulación importante post-transcripcional. Sí, claro.
Y, bueno, la pregunta que yo tengo es referente al microRNA. ¿Es el único blanco que se le conoce o que se le ha propuesto a este microRNA?
Hay otros, no hemos estudiado eso mucho, pero sí creo que sí hay otros blancos. Como que si checas este microRNA de hace algunos años, el blanco es uno y después más recientemente el blanco es otro, probablemente tiene varios blancos. Tenemos que checar eso todavía.
Y mi pregunta es si después se vuelve a expresar ese microRNA en alguna otra región de la planta y si se sabe cómo se regula.
Sí, claro, pues nos gustaría expresar el microRNA en el pues no del padre, pero en el cigoto o en el célula huevo. Sí, sería muy interesante de hacer varios experimentos viendo si importa, si realmente tiene que ser heredado de la célula espermática o si lo expresa en el cigoto tiene la misma función. O otro experimento interesante sería de hacer una versión de AIN3 que es resistente al microRNA. Uno pensaría ver el efecto opuesto. Sí. Vamos a contemplar muchos experimentos así.
Bueno, si ya no hay más preguntas, pues muchas gracias.
Muchas gracias.
Por su presentación.